Phylogenetic analysis can be done by using two sort of informations. Number one is morphological processing. Morphological processing क्या है? For example, length of different physical structures such as legs, hands, and then appearance of other things such as wings, which were there in some species and not in the down species, or which have appeared after some time. And similarly, the other thing is basically macromolecular based information, uh, which is based on internal structures such as DNA, RNA, and protein structure molecules. Now what happens is there are two sort of trees which can be generated from this information, from morphological and from macromolecular information. Those trees are species trees and gene trees. So species trees are basically those trees which are based on species which are from the common ancestor. On the other hand, gene trees, they are used or they are made by using information from genes sequences. तो अब इनकी डिटेल हमारे पास क्या है बेसिकली फाइलोजेनेटिक ट्री में हमारे पास क्या होता है देयर आर पैटर्न्स और देयर आर ब्रांचेस ऑफ स्पीशीज विच आर फ्रॉम द सेम लाइनेज ऑफ species. So same lineage means that they are on the similar line or horizontally they are at the same level. Then the other one gene tree, they are basically made or formulated because of gene duplication event. So gene duplication event means that there would be loss or duplication. There are two choices only. So what happens is it is also broken into several pieces due to its recombination with population. So what happens? It combines with population and then it will modify itself positively or negatively. So if I do proper definition of species tree, then it would be something or a tree which has basically relationship among species that are believed from a that are believed to have a common ancestor. So here I have given an example of frog, mouse, or human, and using species tree where these are all three nodes of a common tree, where there is a tet is a root node, and then we have frog, human, and mouse, so which are different nodes for a species tree. So what is happening here? We are just showing name of the species, and then we are not just showing what is the relationship between these. The relationship means how one thing is changing into the other. But in gene information, what we say that how things have evolved, and this evolution is basically because of duplication of DNA sequences or because of loss of DNA sequences, or which are called gaps also. So if I continue, here I have shown again the example of frog, human, and mouse, and then here I have also shown information that this change is basically because of this genes change. So gene 1 of frog, gene 2 of frog, so this is different from gene of mouse. So what has happened is in the species tree we only had this frog, this mouse, and then this human. But here what we have added is we have also added gene information. So you can also see that there are different levels of these genes. For example, for frog we have two genes. For humans we also have two genes. So gene is duplicated here, and might be in some cases there would be loss of genes also. So nodes are shown as speciation nodes where this information or this weight of species is occurring. And here I have shown this species which are frog and human. So what are homologs now? Now we have seen what are gene tree and what are species tree, and we will see that what is application of gene trees. So basically there is one concept called homolog. Homologs are basically those gene sequences where one gene is related to another gene and they are from the common ancestral DNA sequence. तो उनका जो common ancestral sequence है DNA का वो common है, और there are two parts, gene 1 and gene 2. Based on this definition, we have two types of these homologs, which are orthologs and paralogs. In orthologs, what we have a common parent as shown in this diagram, that we have one common parent, this one, and then we have a speciation event which is occurring here, and we name this primates. Primates basically is a common ancestor for chimpanzee and human, and here we have shown these two things. And if I move on, what will happen? After orthologs, we have another thing. This is called paralogs basically. So what is paralogs? Paralogs are genes which are related by duplication within a genome, and they evolve new functions. So what is happening here? That although we have a one common ancestor which is shown here, and then we have primates and rodents. So they are from the common ancestor, but what happened? Now we have different chimpanzee and human which are from the primates part, and we have rat and mouse which are from the rodents part. Although all these four have the common ancestor, which is shown here, but based on their duplication of gene event, they have generated different species, which are shown in the last this part. Okay, so what is gene tree reconstruction? Now we have sequences of gene and we want to construct a tree out of that thing. We have two choices. Number one choice is microevolutionary information, which considers only sequence information, and the other one is macromolecular or macroevolutionary information, which considers evolutionary plus loss and gain information also, which is duplication event. Although first one is more efficiently or more is used most of the times as compared to the second one. And finally, what is the conclusion? That we have gene tree and sequence tree. Gene tree is more concerned or more helpful for finding information or relatedness amongst genes which may change across species throughout the tree of life.